We've been talking about proteins and that both prokaryotic cells, bacteria, and eukaryotic cells need proteins. So, when does a cell know when exactly to make proteins and enzymes? Have you ever gone to the store and you purchased something that you thought that you needed, and then when you got it home, you realized that you had many of those items, but then you also didn't get the item that you really needed? It's really frustrating because now you've spent money and time buying something that you don't need. The cell is very similar to that. The cell is not going to spend a lot of time, energy, and resources into making something that it doesn't need. This is the same type of thing in the business world. Best Buy is not going to be stocking a lot of PlayStations if it has a warehouse full of PlayStations. So the cell is a little bit like that supply and demand. A cell is only going to make proteins and enzymes when they're actually needed. Otherwise, it's going to spend some valuable resources when it doesn't need to. And sometimes those products can actually build up to be waste, and that's not going to be good for the cell because that could actually be toxic to it. So, bacteria, prokaryotic cells, have a way to actually control when they make enzymes and when they make proteins. And this happens at the transcriptional level. So, when we talked about protein synthesis, we talked about how the DNA contains the code, the genes, to make the protein. And then the cell would make a copy of that gene and keep it in the form of messenger RNA. So when bacteria are going through transcription, they can actually control when the genes are expressed and when the protein and the enzyme are made. So if you take a look at this screen right here, this is actually a picture of prokaryotic DNA. You notice that there are the structural genes. So that is going to code for a protein or an enzyme. And then we also have the P, the promoter. That's where the RNA polymerase is going to bind. What's a little different in the prokaryotic cell is that you notice next to the promoter, there is an operator. So that little purple O region, that's the operator. So there's something called a repressor protein that can actually bind to the operator. So if that repressor protein is bound to the operator, when RNA polymerase binds to that promoter region, it's going to be blocked. It's almost like something blocking a train track. And so the train is not going to be able to go through. If you take a look at that I region, so that yellow region before the promoter, that's a regulatory gene. That actually codes for the repressor protein. So the way that prokaryotic cells can control when they make proteins and enzymes is whether or not a repressor is bound to that operator. If a repressor is bound to the operator, when RNA polymerase binds, it's not going to be able to transcribe the genes. When the repressor is not bound to the operator, then when RNA polymerase binds to the promoter region, transcription and ultimately translation and the making of that protein or enzyme can happen. So I'm going to talk to you a little bit about a certain operon. That is the lac operon. And the lac operon actually codes for lactase. And lactase is an enzyme, and it actually works on a disaccharide lactose. So we know that enzymes usually are named for the substrates they work on, and they end in ASE. So lactose is a disaccharide, and lactase, the enzyme, is kind of like molecular scissors that will break apart that disaccharide into its two monosaccharides, glucose and galactose. You're probably familiar with lactose because this is what's found in milk. So this is a milk sugar. And people that are lactose intolerant actually lack enough of the lactase enzyme. Well, bacteria also make lactase because when lactose is available to them, they need that lactase to break it down so that they can make that monosaccharide glucose. If you kind of think about lactose as being sort of like broccoli for the bacteria, if you're really hungry, you're going to eat broccoli. But personally, I'd rather have chocolate than broccoli. And so glucose is sort of like chocolate for the bacteria. So it can do kind of this magic act where it can actually take that broccoli and then convert it into that chocolate. So I think you can agree that it's probably more desirable. I would rather have chocolate than broccoli. So this is exactly what the bacteria wants to do. It wants to break up that disaccharide so that it can actually use that monosaccharide. So this is how the lac operon works. So again, this is going to be on the transcriptional level. So if you notice these two pictures, we have DNA. Remember, we have the structural genes. So if you take a look at the LAC-Z, LAC-Y, and LAC-A, those are going to be the structural genes that are going to code for lactase. Now, we need a builder to actually build that message. That's going to be RNA polymerase. RNA polymerase binds to the promoter region, and then it can transcribe those genes. Well, I just mentioned the repressor protein. A repressor protein can actually block transcription. So again, the way that this repressor works and this gene expression is based on need of the cell. If the cell has lactose, it wants to break that lactose down. It wants to convert that broccoli into chocolate. So if lactose is available, then the cell is going to need that lactase, that enzyme, to actually break down that lactose. If lactose is not there, why bother spending the time, energy, and resources into making that enzyme that the cell doesn't need? So this is what is actually happening in each of these pictures. So take a look at the top picture. So again, this is DNA in the absence of lactose. So the cell does not have that disaccharide, so there's no need to make lactase to break it down. So in this case, the repressor is actually bound to the operator. And you can see that gray repressor protein bound to the operator. So even when RNA polymerase binds to the promoter region, it's blocked and it can't continue with transcription. If transcription is blocked, ultimately translation will be blocked and that enzyme won't be made. The bottom picture is showing us when there's the presence of lactose. So when lactose is available, the bacteria wants to break it down so it can use it and it can get that monosaccharide, it can get that glucose. So when lactose is available, the presence of lactose will basically bind to that repressor and pull it off of the operator region. So the lactose will bind to repressor, take it off of the DNA, and now when RNA polymerase binds to the promoter region, now the DNA is free and clear so that now transcription can happen. So again, this is a lot like supply and demand. When that disaccharide is available, it needs the enzyme to break it down. So by either putting that repressor on the operator or taking it off, it's able to control that gene expression and when it makes that lactase. So again, this is a lot like supply and demand. So oftentimes what I do is I like to go through and I like to ask myself questions. Okay, does the cell need it? When is the enzyme going to be made? So I've kind of posed a couple of questions here. And again, think about it from that supply and demand. When is it needed? When is it not needed? So if we take a look at our first questions, if lactose levels are high, do we need lactase? Does the cell need to make lactase? Yes, because lactose is there, so we need to break it down. Where is the repressor bound? Remember, lactose, that presence, is going to pull off that repressor. So the repressor is actually bound to the lactose. So can transcription happen? Yes, DNA is not blocked. So when RNA polymerase binds to that promoter region, transcription can happen, and ultimately that lactase will be made. If lactose levels are low, does the cell need to make lactase? No, there's no need for it because there's no lactose to break down. So in that case, where is the repressor bound? The repressor is then going to be bound to the operator. Can transcription happen? No, DNA is blocked because that repressor protein is bound to the operator. So even when RNA polymerase binds to the promoter region, it's kind of like something is blocking that train track. That repressor protein is a big obstacle, so transcription can't happen. Ultimately, that lactase won't be made. So it's like that supply and demand. When does the cell need it? When does it not need it? Now, I know that it can be a little abstract to be thinking about lactose and when a bacteria needs to make this enzyme. So sometimes what I do is I think of something that's a little more relatable. So if I was thinking about maybe guacamole and chips, I love to eat guacamole and chips. So if I don't have chips, do I need to make guacamole? So, no, because I don't have anything to eat the guacamole with. So, you can take whatever it is that you can relate to and something that maybe that you like. When do you need it? When do you not need it? And that can kind of help you to understand when the cell needs to make product and when it doesn't.